Hi everyone, we are the Group 5 and today we will be presenting our research titled Fermentation of Sugars, the Role of Carbohydrates in Biofuel Production and Sustainable Energy Solutions. I am Esther Bell L. Acot.
My name is July C. Gabunada.
I am Rodel Guillermo Jr.
I am Michael Arsha.
I'm Hannah Rochelle Sarti.
I am Jesselyn Torres.
Carbohydrates and their significance in biochemistry. Carbohydrates are essential biomolecules composed of carbon, hydrogen, and oxygen, typically adhering to the general formula CH2O. They are broadly categorized into monosaccharides, disaccharides, and polysaccharides according to Khan Academy 2021. Owing to their structural diversity and chemical reactivity, carbohydrates are integral to a wide array of biological functions. As noted by Batla and Lau 2023, carbohydrates serve as the principal energy source in living organisms. Glucose provides an immediate energy supply, while storage polysaccharides such as glycogen and starch function as energy reserves. Beyond their metabolic roles, carbohydrates contribute to cellular recognition, signaling, and structural integrity. The term encompasses not only simple sugars, but also complex forms like oligosaccharides and polysaccharides according to Benozik et al. 2020. Of particular interest is their environmental abundance. Cellulose and starch, for instance, are plant-derived carbohydrates composed of polymerized glucose units and are among the most ubiquitous organic compounds on Earth, according to Chandal 2021. This biochemical versatility makes carbohydrate a compelling focus for research in sustainable energy conversion. This is relevant to our chosen topic as the global demand for clean and renewable energy intensifies. The role of carbohydrates in bioethanol production has become increasingly significant. Unlike fossil fuels, biofuels derived from fermentable sugars offer reduced greenhouse gas emission, mitigate reliance on finite energy sources, and promote rural economic development according to NAF 2024. Also, according to Getachew-Demi et al. 2023, substrates rich in fermentable sugars such as food waste present in viable feedstocks for bioethanol fermentation. Supporting this claim, Aegis et al. 2024 emphasized the synergy benefit of aligning biofuel generation with waste management initiatives, thereby addressing both energy and environmental challenges. Additionally, Kasmi et al. 2025 underscored the necessity of safeguarding food security and ecological balance while scaling up bioethanol technologies. The research gap of this study is that despite ongoing advancement, a knowledge gap persists in understanding how different carbohydrate sources and microbial fermentation strategies influence the sustainability, efficiency, and environmental outcomes of biofuel production. Existing studies often focus on isolated aspects, such as organism optimization or feedstock processing, but lack an integrated assessment of biochemical, ecological, and economic dimensions. This study sought to bridge the aforementioned gap by examining the role of carbohydrates in sugar fermentation and their broader implications for bioethanol-based sustainable energy.
Research objectives. Our study focuses on the role of carbohydrates in sugar fermentation for biofuel production. The research objectives were carefully chosen to explore this topic from multiple angles: biochemical, microbial, and environmental. The first objective is to classify the most commonly used carbohydrate types in biofuel fermentation and analyze their biochemical properties. This is important because carbohydrates like glucose, sucrose, and cellulose behave differently during fermentation. Understanding their properties helps identify which ones are most efficient for ethanol production. The second objective is to analyze the efficiency of various microorganisms in fermenting different sugars. Different microbes have different abilities. For example, yeast can quickly ferment simple sugars, but complex sugars from biomass need special microbes or treatment. This helps improve fermentation strategies. The third objective is to assess the long-term sustainability of carbohydrate-based biofuels in terms of energy output, resource availability, and cost. This is essential for determining whether biofuels can truly replace fossil fuels in the future, not just technically, but practically. The fourth and last objective is to evaluate the environmental impact of sugar-based biofuel production, especially regarding carbon emissions and ecological balance. Biofuels are often considered clean, but they can also affect land use, water, and biodiversity. So by addressing these objectives, this study would provide valuable insights into carbohydrate-based biofuel production, supporting the transition to cleaner and more sustainable energy solutions. Understanding sugar fermentation, microbial efficiency, and environmental impact will help refine biofuel strategies while balancing technological progress and ecological responsibility. Now let's move on to Chapter 3, Discussion and Analysis. Classification and biochemical properties of carbohydrates in fermentation. Carbohydrates play a vital role in bioethanol production. They are classified into monosaccharides, disaccharides, and polysaccharides, and each class behaves differently during fermentation. Monosaccharides, like glucose and fructose, are simple sugars. According to Peters 2006, glucose is easily metabolized by ethanol-producing microorganisms like Saccharomyces cerevisiae. Because of their simple unbranched structure, these sugars are absorbed and fermented with high efficiency. Disaccharides, such as sucrose from sugarcane, must first be hydrolyzed into glucose and fructose before fermentation. Despite this extra step, they are still highly effective for ethanol production as supported by Niaz et al. 2020. Polysaccharides, including cellulose and hemicellulose, are more complex. These are found in lignocellulosic biomass and required advanced pretreatment processes and specialized enzymes for conversion into fermentable sugars. This makes them more challenging but also promising for second generation biofuel. Next is the analysis of their fermentability and biochemical significance in ethanol production. The structure of each carbohydrate influences its fermentability. Glucose and fructose are readily fermented and offer high yields. Sucrose requires enzymatic breakdown, but is still widely used. Cellulose and hemicellulose require hydrolysis, making the process more energy intensive. Despite these challenges, complex carbohydrates are abundant in biomass waste, which supports environmental sustainability. Onay and Supriya et al. Note that first generation biofuels from corn and sugarcane dominated today due to ease and efficiency. However, Xiao et al. 2024 highlighted how genetically engineered microbes now allow us to ferment mixed sugars and tolerate inhibitors, closing the gap between first and second generation biofuels. Role of carbohydrate structure in fermentation efficiency. The molecular structure of carbohydrates significantly affects their fermentative behavior. Simple sugars like glucose are directly fermentable by most microbes. Complex sugar require enzymatic treatment before microbial uptake. Niaz et al. 2020 stated that fermentable carbohydrates are present in many agricultural feedstock like rice, wheat, sugarcane, vegetables, fruits, and dairy. Online 2021 also emphasizes non-traditional sources like algae, food waste, and cyanobacteria, which are rich in starch and cellulose. Matching the carbohydrate structure with the microbial system is critical. The wrong pairing could reduce yield and efficiency, while the right one can optimize the process and make it more cost-effective and sustainable. Mechanisms of microbial fermentation and sugar breakdown. Microbial fermentation is a core process that transforms sugars into ethanol. According to Brown et al. 2024, corn and sugarcane sugars are the primary substrates in first-generation bioethanol production. Microbes like Saccharomyces cerevisiae convert glucose into ethanol rapidly under anaerobic conditions. However, with complex carbohydrates such as cellulose, pretreatment is necessary to break down the long chains into fermentable monosaccharides. This step is often expensive and technically demanding. Fallars et al. 2018 emphasized that lignocellulosic biomass, while sustainable, possesses a challenge due to its resistance to microbial digestion. As a result, researchers are working to develop adapted microbial strains that can handle these materials more efficiently. Brown et al. Also advocate for sustainable feedstock strategies that don't compete with food sources.
I will be discussing the microbial efficiency in fermenting sugars, comparative analysis of yeast versus bacteria efficiency in bioethanol production. Yeast, especially Saccharomyces cerevisiae, are widely used in industrial ethanol production. For their efficient conversion of hexes under anaerobic conditions. Tufalo et al. 2019 recombinant strains further enhance sugar uptake and tolerance to fermentations inhibitors, boosting efficiency. Zhao et al. 2024, on the other hand, bacteria like Lab and AAB follow different pathways, producing lactic and lactic acid, making them more relevant to food processing. Then fuel, Wang et al. 2022 and Han et al. 2024. The microbial efficiency in fermenting sugars, factors affecting microbial activity including pH, temperature, and sugar concentration. Fermentation efficiency depends on tightly controlled peel and temperature, while ensure microbial health and enzyme activity. Recombinant S. Reservisae strains have higher ethanol yields under optimal setting to enhance sugar metabolism and also in inhibitor tolerance. Zhao et al. 2024, even slight divisions can cause stress and reduce efficiency. Fermentations inhibitors such as furans, organic acids, and phenolic compounds formed during biomass hydrolysis disrupt cell members and enzyme function. And let's proceed to the number 3 of the discussion, the sustainability and feasibility of carbohydrates on biofuel, based biofuels. Advantages and challenges of carbohydrates derived biofuels versus petroleum-based fuels. Carbohydrate-based biofuels are gaining global interest as sustainable alternatives to fossil fuels. Unlike fossil fuels, carbohydrates are renewable and regenerate quickly. Plant-derived carbohydrates and organic waste are abundant energy sources for advanced biofuels. Hawang et al. 2022. A key example is 2,5-dimethylfuran or DMF, a biofuel synthesized from waste carbohydrates. DMF has fuel properties similar to petroleum and supple in current engineers without major changes. Advances in catalyst engineering are improving its commercial potential and beyond terrestrial biomass. Cyanobacteria are emerging as a promising biofuel sources. The economic and environmental feasibility of large-scale bioethanol production. Large-scale bioethanol production is shaped by both economic and environmental factors. In India, Nande et al. 2023 used a MILP model in Punjab, showing that residues like paddy and wet straw can meet sustainability goals under supportive policies. However, global deployment still faces financial, infrastructural, and policy challenges. Second-generation feedstocks ace food versus fuel concerns but demand advanced and costly technology. Perrin and Jones 2019 highlighted microalgae as a promising biofuel source, though it's not yet economically mature. Long-term sustainability concerns and potential biotechnical improvements. Despite being renewable, first-generation biofuels raise sustainability concerns. Large-scale cultivation crops like corn can divert land. Water and labor from food production worsening food insecurity. Jeswani et al. In 2020, expansion of agricultural land may also drive deforestation and ecological disruption. Subramaniam et al. In 2020, fertilizer and pesticide use further contribute to soil degradation and water pollution. Still, biofuels offer cleaner energy option with lower emissions, and sectors like transport and industry. Chowdhury et al. In 2025, sustainable use requires a move towards second-generation feedstocks and improvement land management.
So today, I'm going to continue to discuss the environmental impact of sugar-based biofuel production, carbon emissions and ecological footprints of fermentation-based biofuels. So sugar-based biofuels like methanol can significantly reduce GHG emissions. Ethanol from sweet sorghum juice can lower sweet oils emissions by up to 72% when vinasse is reused as fertilizer, according to Shi et al. 2022. According to Morrissey et al. 2021, found a 48% decrease in climate impact and 52% reduction in fossil fuel use from sorghum-based biofuels. So, however, 1G biofuels raise concerns over land use change, deforestation, water scarcity, and nutrient run runoff according to Jeswani et al. 2020 and Subramaniam et al. 2020. So intensive farming can cause soil erosion and disrupt ecosystem according to Sivara Nakumar et al. 2023. The strategy for reducing environmental impact and enhancing biofuel efficiency. So to reduce the environmental of 1G, sugar-based biofuels strategic measures are essential. Food stocks like corn and sugarcane compete with food crops for land, water, and labor, according to Jeswane et al. 2022, while increase cultivation and accelerate deforestation and land conversion, according to Sobra Neyam et al. 2020. So excessive use of fertilizer and pesticide leads to soil degradation, eutrophication, and freshwater pollution, according to Jeswane et al. 2020. However, when sustainably managed biofuel offer cleaner alternatives and reduce life cycles emissions according to Kaudhari et al. 2025. So next, the future trends in biofuel refinement and environmental sustainability. So innovations in nanotechnology, metabolic engineering, AI, and adaptive system are enhancing biofuel efficiency and environmental performance. These tools improve conversion rates and boost sustainability. According to Kadhari et al. 2025, noted that future success depends on cross-sector coordination, aligning biofuels with global goals like UN SDGs. These involve creating standardized assessment, sustainability, benchmarks, and strong policy support that addresses ecological and socio-economic impacts. Ultimately, while sugar-based biofuels help decarbonize energy, their sustainability depends on cleaner tech, diverse food stocks, and responsible land use, ensuring long-term environmental and economic balance. Therefore, summary of findings. First, recap of classified carbohydrate types and their biochemical role in fermentation. Carbohydrate play a fundamental role in biofuel fermentation, serving as the primary source of fermentable sugars. They are classified into monosaccharides like glucose and fructose, disaccharides like sucrose, and polysaccharides like starch and cellulose, each contributing uniquely to ethanol production. Monosaccharides are readily fermented by microorganisms due to their simple structure, while disaccharides like sucrose require enzymatic hydrolysis prior to fermentation. Polysaccharides, particularly those found in lignocellulosic biomass, necessitate complex pre-treatment processes to release fermentable monomers, making them more technically demanding but crucial for second generation's biofuel. Second is the overview of microbial efficiency in sugar conversion to biofuel. So macroorganisms such as yeast and Zymomonas mobilis are widely used for fermenting simple carbohydrates due to their high ethanol yields and metabolic efficiency. However, converting complex sugars from lignocellulosic biomass remain more challenging. Microbial efficiency is largely influenced by two factors: the ability to tolerate high concentrations of sugars and ethanol, and the metabolic capacity to utilize diverse sugar types, including glucose, xylose, and arabinose. Genetically engineered strains have improved performance in these conditions, contributing to more efficient second and third generation biofuel processes.
Chapter 4, Summary of Findings. Summary of Sustainability Assessments for Carbohydrate-Based Biofuels. Carbohydrate-based biofuels offers a renewable alternative to fossil fuels but raised sustainability concerns related to land use, food security, and resource competition. First generation biofuels derived from sugarcane and corn are commercially viable yet criticized for their environmental and socio-economic impacts. In contrast, second generation biofuels produced from agricultural residues, lignocellulosic feedstocks, and cyanobacteria present a more sustainable option. However, realizing their full potential requires continued advances in technology to enhance conversion efficiency and reduce production costs. Insight into environmental impacts and potential mitigation strategies. While sugar-based biofuels reduce greenhouse gas emission relative to fossil fuels, they also pose environmental risks. Land use changes, water overuse, and excessive reliance on fertilizers and pesticides can lead to deforestation, soil degradation, and biodiversity loss. Furthermore, food versus fuel concerns persist, particularly in regions with limited agricultural resources. Mitigation strategies include shifting to non-food feedstocks such as crop residues, microalgae, and organic waste, as well as integrating phytoremediation to rehabilitate degraded lands. Technological innovation such as nanotechnology, smart fermentation systems, and AI-driven optimization also show promise in reducing the ecological footprint of biofuel production and supporting long-term sustainability. Chapter 4 Conclusion. This study has illuminated the critical role in carbohydrate in the fermentation process of bioethanol production, emphasizing their biochemical diversity, microbial interactions, and sustainability implication through the classification of carbohydrate types, monosaccharides, disaccharides, and polysaccharides. It has become clear that their structural complexity directly influences fermentation, efficiency, and sustainability as biofuel feedstocks. While simple sugar offer high yield and ease of conversion, polysaccharide-rich lignocellulosic biomass presents a more sustainable, through technologically demanding alternative. The analysis also highlighted the importance of microbial efficiency in sugar metabolism, particularly focusing on the dominance of Saccharomyces in ethanol fermentation and the emerging potential of genetically engineered strains, variability in sugar composition, environmental conditions, and inhibitor presence requires adaptive microbial strategies to optimize conversion rates. In the terms of sustainability, carbohydrate-derived biofuels present both promise and paradox. On one hand, they contribute to greenhouse gas reduction and energy diversification. On the other, they raise concerns regarding land use, food security, and ecological degradation. Mitigation strategies, including the adoption of second-generation feedstocks, integrated waste valorization, and emerging technologies, offer a path forward, aligning biofuel development with broader environmental and economic objectives. Ultimately, this research underscores the necessity of a balanced, multidisciplinary approach in advancing carbohydrate-based biofuels by integrating biochemical innovation, microbial optimization, and ecological responsibility. Bioethanol production can evolve into Bioethanol production can evolve into a cornerstone of sustainable energy systems, fueling not only engines but also global pursuit of cleaner and more equity.
Chapter 4 Recommendations. Advanced research on polysaccharide conversion technologies. This means you should continued exploration of pretreatment and enzymatic hydrolysis techniques is essential to unlock the potential of complex polysaccharides, such as cellulose and hemicellulose. And improving these processes will elevate the efficiency and feasibility of second-generation bioethanol production. Next is enhanced microbial engineering for broader sugar utilization. This means developing genetically optimized microorganisms with increased sugar uptake capabilities and tolerance to fermentation inhibitors will strengthen microbial adaptability, enabling efficient conversion of diverse carbohydrate sources, including pentoses from lignocellulosic biomass. Third is promote sustainable feedstock alternatives. This means to alleviate land use pressures and food competition. It is recommended to prioritize non-food biomass, such as agricultural residues, cyanobacteria, and microalgae feedstocks for biofuel production, and these options present lower environmental trade-offs and support long-term resource security. Fourth is integrate environmental safeguards into biofuel systems. This means comprehensive environmental assessments and the application of circular resource management practices such as phytoremediation are critical for minimizing ecological harm, and these safeguards ensure that biofuel development aligns with biodiversity protection, water conservation, and soil health. Fifth is support innovation through policy and infrastructure. This means robust policy support and investment in infrastructure are vital for scaling biofuel technologies, and governments and institutions should foster research funding, establish sustainability guidelines, and improve biomass logistics to support rural development and energy resilience. And lastly, foster interdisciplinary collaboration and public awareness. This means greater collaboration between researchers, industry, practitioners, and policymakers will accelerate innovations, and additionally, increasing public awareness about the environmental and economic benefits of carbohydrates-based biofuels can drive informed decision-making and societal acceptance. Next is acknowledgements. First and foremost, we thank God Almighty for giving us strength, guidance, and wisdom throughout the completion of this research study. We are deeply grateful to our AG26 instructor, Ms. Maljoan B. Dizan, for the support, valuable feedback, and encouragement from the beginning until the final output. We thank our family and loved ones for their endless support, prayers, and understanding during times of difficulty. Special thanks to our groupmates and classmates who contributed ideas, time, and teamwork in making this paper possible. Lastly, we acknowledge the authors and sources we have cited, whose work helped us in building the content of this research.